All right, so thank you all for tuning in today. I'm Dr. Hunter Frame. I'm the field crops agronomist here at Tidewater Agriculture Research and Extension Center with Virginia Tech in Suffolk, Virginia. We were going to do a video today out in the field. However, Mother Nature has dropped about 1 inch to 2 inches of rain on us the past 3 or 4 days, so the fields are saturated. So what we're going to do is give a little bit of a tour and an overview of what my soil fertility nutrient management lab is doing during weather delays, but also in the wintertime. We process a lot of samples, a lot of soil samples, plant tissue samples in this lab, especially for nitrogen management in cotton and corn systems. And so stay tuned, and we're going to go through some of the things that we do that are pretty unique to our program and pretty unique to nitrogen management and cropping systems. As you can see from the equipment behind me, this is a wet chemistry lab. We work mainly with nutrient extractions from soils and plant tissues. We have very basic equipment in terms of balances here. We have a 2-place balance, 4-place balance for weighing out chemicals for reagents for our auto analyzers, as well as weighing out samples of fertilizer, which we'll get into in a little bit later. We have all of our glassware, ovens, fume hoods for safety, but the workhorses of our lab, as we get into the lab, the first workhorse of our lab is this analyzer right here. And it doesn't look like much when it's not running. We have auto sampler, and then we have the latchet flow injection colorimeter, where we analyze samples for nitrate, ammonium, phosphorus, total nitrogen, total phosphorus. We could also do petiole nitrates on this machine. This machine annually runs anywhere from 7,000 to 10,000 samples across it a year in my lab, ranging from plant tissue to soil extractions. And it's our workhorse in terms of analytical equipment. We also have a dry combustion analyzer in a second lab that we have, where we analyze plant tissues and soils for percent carbon, nitrogen, and sulfur analysis. The next workhorse that we have in our lab is this system. These boxes is set of 3 boxes here, and we actually have 2 of these systems. This is our smaller ammonia volatilization chamber set. We can run 6 treatments replicated 3 times. These are temperature controlled, humidity controlled, and the airflow is controlled. In these boxes, we measure ammonia loss from surface applied or subsurface applied fertilizers treated with and without different nitrogen stabilizers. The second set we have is a little bit larger. We can do 9 treatments replicated 3 times. Okay. So here we have the basic components of our ammonia volatilization system. If you look in the box, we have 6 chambers that are set up. We can put different soil types in the chambers. These are the chambers, the white caps, and we have internal water columns with the black cap columns here. We have a fan and a heater strip and then a thermostat to control the temperature and airflow to make sure the airflow is constantly circulated in these boxes. Now, what's the point of all of this? So here we have an external water column. So the water columns help bring humidity up in the airstream that's continuously pumped through these chambers. So we bring the humidity up with the outside water column, as well as it helps scrub the air of any ambient ammonium that's in the air. Then the airflow comes out of the external water columns into the internal water columns, which are inside the temperature control cabinet, and that brings the air up to close to 100% humidity inside the chamber. This does 2 things. It further scrubs the air of ammonia, but it humidifies it so not to dry our soil out, which is in our chamber. Now, these are specialty designed chambers, and in it we can put multiple soil types, and we adjust the soil moisture accordingly. So typically, a typical trial, we'll run the soil moisture at 2/3 field capacity, and field capacity will be determined using a pressure plate apparatus, which is another piece of soil analytical equipment, and we'll adjust that based on the soil type. So all soils that we test have a particle size analysis, so we know the texture, we know the chemical characteristics as well from a soil test, but we also do water holding capacity. Once the airflow enters the chamber through a downpipe, circulated in the chamber, and this chamber volume is replaced about every 30 seconds, the air inside of this chamber. It comes further out of the chamber and then flows into our acid trap. Now, this is dilute phosphoric acid, and this captures any ammonia in the air. So when the ammonia reacts with the acid in this bottle, it converts it to ammonium, and it's trapped. From there, the acid bottles will be analyzed on our flow injection colorimeter. for analysis to see how much ammonia we actually lost. And I'll go through a chart of some of the experiments that we have ongoing, looking at different nitrogen placements as well as enhanced efficiency fertilizers to combat ammonia loss in our crop fields. All right, so some of the products and some of the things that we test are actually commercial products that are available on the market. So any mention of trade names is not an endorsement of the product, but it's a current standard inhibitor that we're testing for a protocol that's funded by the Fertilizer Institute through their 4R grant that we received last year, and it's funded for a 5-year project. So we can test different fertilizer materials, and here we have a granular fertilizer that has been treated with NBPT or a product called Anvil, and this is a product that inhibits ammonia volatilization using the molecule NBPT, and once it's treated, this will slow down transformation of urea to ammonium in the soil. Second product is another granular product we're using. It's actually a nitrification inhibitor called Instinct 2, where the active ingredient is nitropyrin. And this product is a nitrification inhibitor. However, it does have some effects, maybe adverse effects, on ammonia volatilization, so we're testing that in the chambers. And then we can also test liquid fertilizer formulations. This is UAN that has been treated with Anvil, as you can see the green color. UAN treated with Instinct, the milky color here. And then UAN with the dual inhibitors in it, so Anvil and Instinct. And this is, we're looking at how do these inhibitors play off of one another, what do they do in terms of stabilizing the nitrogen forms in the soil, what effect that has on ammonia volatilization. And so you're probably asking, well, this is all great science, but what does this have to do with farming and production agriculture? Well, with products like the NBPT-based products that are on the market for ammonia volatilization, we've measured as high as an 88 bushel yield advantage using those products simply because of the nitrogen that is stabilized and not lost as ammonia to the atmosphere. That's a significant financial savings for the farmer, not in terms of just cost of nitrogen, but also in terms of the increased yield per unit of nitrogen that we see, or increased nitrogen use efficiency in that cropping system. So these products, when used properly and used in environments and in soil types that will benefit producers the most, can be advantageous economically as well as efficiency wise in terms of limiting the amount of environmental loss of nitrogen in the ecosystems. And that's why we're testing them in the lab, so we can help screen practices and products that are the most beneficial and scale that up to the field setting in our field plots moving forward to help farmers not only in Virginia but across the country. We're testing soils all the way from Arizona to Virginia, down to Alabama, Georgia, all the way up to Wisconsin in this lab through various projects. And with that, I'd like to thank you for your time and you watching this video and learning a little bit more about the Virginia Tech Soil Fertility and Nutrient Management Cotton Lab at Tidewater Agriculture Research and Extension Center. Thank you.